All right, we're still in chapter 21. I believe this is the 5th video, and this one is going to be on antibodies, only on antibodies. And antibodies are also called immunoglobulins. And what they are is they are proteins that are secreted by plasma cells. Remember that we've been talking about humoral immunity and the B cells, and the B cells differentiate into either plasma cells or memory B cells, and those plasma cells make antibodies, and that's exactly what we're talking about. And this makes up gamma globulin portion of the blood. It's capable of binding specifically with an antigen detected by B cells, and it's going to be grouped into one of 5 Ig classes. So the basic antibody structure, I did not ask about this on the exam, just so you know, but I do want you to know that it either looks like a T or a Y shaped antibody monomer consists of 4 looping polypeptide chains linked by disulfide bonds. Okay, so it's 4 chains. 2 of them are identical, and the other 2 are also identical. And so there's 2 heavy chains and 2 light chains. And this variable V region at one end of the arm combined to form 2 identical antigen binding sites. Then there's the stems that make up the constant region. This is the area that determines the antibody class, and this is considered the effector region of the antibody. And it serves common functions in all antibodies, dictating the type of cells and/or chemicals that the antibody can bind and how the antibody class functions to eliminate antigens. So, for example, some antibodies fix complement, some circulate in the blood, some are found in body secretions. And so here we have this kind of Y-shaped structure. This would be the antibody monomer. We have the heavy chain, identical to that heavy chain, and a light chain, identical to that light chain. The antigen binding site would be here. And this is also antibody structure. All right, so the 5 major classes of antibodies are IgM, IgA, IgD, IgG, and IgE. IgM is a pentamer. Pent means 5. It's going to be larger than the others. It's the first antibody released. It is a potent agglutinating agent. I don't know if you remember talking about agglutination back in Unit 1. We talked about it when we were talking about blood agglutinating, where if you end up getting somebody else's mismatched blood type, then agglutination can occur, and that's clumping. And this is readily fixes and activates complement. Then we have IgA, or secretory IgA. This is a monomer or dimer. It's found in mucus and other secretions, and this is going to help prevent the entry of pathogens. So here's our pentamer, IgM, 1, 2, 3, 4, 5, and you can see they're all linked together. And then we have IgA. This is a dimer, so we see 1, 2. You do not have to know all the extra stuff that's on here, okay? I just want you to know what was on this slide right here. We have IgD. This is a monomer attached to the surface of B cells, and it functions as a B cell receptor. IgG is a monomer. 75 to 85% of antibodies in plasma. This is going to, that was supposed to say form. Maybe not, maybe from, I don't know. Anyway, it's the antibody that you see for secondary responses and late primary responses, and it crosses the placental barrier. And then there's IgE, and monomer active in some allergies and parasitic infections, and it causes mast cells and basophils to release histamine. And so here are these guys. We see IgD is a monomer. These are all monomers. Monomer, monomer, monomer. So plasma B cells can switch from making one class of antibody to another. Now, it does retain its specificity for the same antigen. It's just going to be making a different class of antibody for that same antigen. IgM is released during the primary response, but then the plasma cells can switch to IgG for the secondary response. And then almost all secondary responses are IgG, and then switching from IgM to IgA or IgE can also occur. Antibody targets and functions. So what antibodies do is they inactivate and tag them. They do not destroy antigens. They form antigen-antibody immune complexes. And then there are these 4 defensive mechanisms used by antibodies that we're going to discuss in this chapter: neutralization, agglutination, precipitation, and complement fixation. Neutralization is the simplest one, but probably the most important one of the most important defensive mechanisms. Antibodies just block specific sites on the viruses or bacterial exotoxins, and it just prevents the antigen from binding to receptors on tissue cells. That's it. And then the antigen antibody complexes undergo phagocytosis. Agglutination, again, we talked about this a moment ago, clumping of mismatched blood cells, but antibodies can have the same determinant on 2 different antigens at the same time. And what this does is it allows for antigen antibody complexes to become cross-linked into large lattice-like clumps, called agglutination. Precipitation is similar to agglutination, but it's molecules instead of cells that are going to be cross-linked into complexes, and then the complexes precipitate out of solution. The precipitated complexes are easier for phagocytes to engulf. And then we have complement fixation and activation. The main antibody defense against cellular antigens, bacteria, mismatched red blood cells. When several antibodies are bound close together on the same antigen, complement binding sites on their stem regions are aligned. That may not make a lot of sense, and I can't really draw it in my current circumstance right now. But alignment triggers complement fixation, which leads to cell lysis as well as other complement functions. And an example is the amplification of the inflammatory response and opsonization. So antigen, antibody, and we have inactivates by neutralization, agglutination, or precipitation, and it can also trigger complement activation. And then you can see complement will lead to cell lysis, and it enhances phagocytosis and inflammation, and all these other enhance phagocytosis. Summary of antibody actions. Antigen antibody complexes do not destroy antigens. They prepare them for destruction by innate defenses. Excuse me. Antibodies go after extracellular pathogens. They do not invade solid tissue unless a lesion is present. There is a recent exception that has been found, and that is that antibodies can act intracellularly if they are attached to a virus before it enters the cell, and then it activates mechanisms that destroy the virus. And here's your question. You guys have a good day.